Okay, so now we are going to look at the peptide bond formation. So polypeptide are linear polymers of an amino acid linked by peptide bonds, or we call it an amide linkage. And this amide linkage formed by nucleophilic acyl substitution. That is, the N-terminal amino acid has the free amino group, and the C terminal has a free carboxylic group. So therefore, there is a dehydration reaction that will take place. That is, it means that in this case, the N-terminal amino acids of the amino group and the C-terminal has the free carboxylic acid, which now linked up together. Like, for example, look at it here. This is a carboxylic group and this is the amino group. And they will now link up together with the dehydration through the dehydration reaction. And when we say dehydration reaction, it means the removal of water. It means the removal of water molecule. So that is the dehydration reaction, and these resulting in amino acid residues and are named by the number of the amino acids. So because here we have one amino acid here joined with another amino acid, making two amino acid, then we now have what we call a dipeptide, because the amino acid are two, and that is why we call it dipeptides. So amino acid sequence leads directly to the protein's native conformation. So now we should understand that in the peptide bond formation is when you have a polypeptide and then linked through a peptide bonds. And in this case, we have what you call the N-terminal amino acid and the C-terminal decarboxylic acid linked together through the hydration reaction. And when we say the hydration reactions means the removal of water molecule, and then we now have the resulting amino acid residue. And then it's also depend on the number of the amino acids residue. So if we have two amino acid is dipeptide, and if we have three, we have tripeptide, and if it is four is tetrapeptides. Okay, and then now we are now going to also look at how some of the properties of this peptide bonds. So peptide bonds generally they are rigid, they are very rigid, and then they are also flat, and they have carbon to nitrogen bond between two amino acids, and which are shorter than the other carbon to nitrogen bond. So it means that they have a bond length. They have a bond length, and this bond length. So therefore, the carbon to nitrogen bonds that are found between two amino acids in a peptide linkage is shorter than that of the normal carbon to nitrogen bond. And the partial double bond characterize the resonance hybrid nature of the peptide. And due to rigidity, one third of the bond in a polypeptide bond cannot freely rotate. So because of the nature of the peptide linkage or the peptide bonds, it is very rigid. So the polypeptide backbone cannot rotate. It cannot rotate because of this rigid linkage, or this rigid amide linkage, or rigid peptide linkage, and that is why it cannot easily be rotated. And then the limits, the number of the conformational possibilities. Okay, then so that is in the case of this. Then we also have we have cysteine oxidation that leads to the reversible disulfide bond. So disulfide bonds are formed when two cysteine residues form this bond. So we have cysteine non-stranded amino acids. It helps to stabilize polypeptide and proteins. So yeah, ladies and gentlemen, we should understand that we have amino acid, and in this we have amino acid called cysteine. And this cysteine can easily form what we call disulfide bridges, because the structure of cysteine, we have what we call thiol groups, which is SH. So we have them. So the linked, or they form a bond, which is what we call disulfide bridges, and this is disulfide bridges. And the presence of disulfide bridges in a proteins, it helps, or yeah, it helps to stabilize the polypeptide and the protein. So it's very, very important. The sulfide bridges, it helps in stabilizing proteins and polypeptides. Okay. And then the peptides. So we have these peptides, they have a biological important role. An example of this peptide, we have glutathione. And glutathione is a tripeptide. It means that it's made of three amino acids, and it's found in almost all living organisms. And the importance of this glutathione is involved in protein and DNA synthesis. So it helps in synthesizing proteins as well as the DNA. So it synthesizes proteins and DNA, and it also helps in metabolizing toxic substances. Like, for example, when we take drugs, drugs is toxic. So we don't want the drugs to stay long in our body. So we need to eliminate it. So in terms of eliminating drugs, we use glutathione as a conjugating agent in removing drugs. And also it's used in the transportation of amino acids. And also, apart from glutathione, we have also vasopressin. So vasopressin is an antidiuretic hormone, and it regulates water balance, appetite, and body temperature. We also have oxytocin, which helps in a signal is a signal peptide. So it aids in uterine contraction during labor, and it also stimulates ejaculation of milk by mammary gland during breastfeeding. And then we also have, apart from this, that is in the case of peptides. So these are the three examples of peptide, and they are very, very important. This is just to show us how important the peptide are. And this peptide, usually in peptide are formed, or are usually between 2 to 50 amino acids. So when you have 2 to 50 amino acids, then of course we have peptides. Like, for example, here in the case of glutathione, it's just 3, so it's formed between 2 to 50. Okay. That is this. And then now the next thing is we are going to look at the protein's diverse function, the functions of proteins. So proteins generally have a diverse function. It's used in catalysis. When we say catalysis, it means it can serve as an enzyme. And remember, an enzymes are substances that speed the rate of biological reaction. So this is a reaction. For example, we have A and B to produce C and D. So we know that usually what actually brings the problems, or what usually causes the problem in, or what led the reaction, is actually when there is no enzyme, because some reactions can take years, hours, days. It can take years, it can take days, and this is due to the presence of this is due to the absence of enzymes. So enzymes are very, very important in speeding the rate of the chemical reaction. So maybe enzymes can combat the reaction that can take a days to make the reactions to happen in just a second. And then also the structure. So proteins also help in the structure and organismal. Like, for example, we have some organism, like in the case of human, we have different cell, and they have different shape. So the shape and the functions, the shape as well as the structure of the particular cell depend on the types of the proteins. Like, for example, we have red blood cells having biconcave shape. So it is biconcave shape because of the presence of proteins that is present in it. And the name of the proteins is hemoglobin. And also it helps in movement. Proteins help in movement. Like in the case of amoeba, they have amoeboid in shape. So the amoeba, they are irregular in shape, and they can move from one point to another. So it's because of the proteins that they have. And it's also have proteins help in the defense system. Like, for example, generally, we have some foreign organisms that can easily get into our body, and we don't want them. They are dangerous. We need to eliminate them. We need to take care, our body need to take care we need to fight them back. So usually some of these proteins, they are antibodies and they help in fighting those kinds of organisms. So it's those kinds of microorganisms that are not needed using the antibodies, and the antibodies are the proteins. It's also regulation. Like for example, it helps. We have insulin. Insulin is a protein that helps to regulate and maintain the level of sugar in our body. Also, in terms of the transport in the membrane of the cells, we have a lot of proteins that help in controlling what get in out of the cell. And storage, we have albumin in bad eggs. Like storage, we have some proteins that their main functions is to actually help in the storage of some materials, and also stress responses. We have heat shock proteins. Sometimes we actually exposed to stress. So we have some proteins that will usually take care of that. Okay. So we also have two main classes of proteins, and these classes of proteins is actually depend on the actually shape of that proteins. And we have fibrous protein and globular proteins, and we also talk on this very well. And then we have the composition. We have the composition of the classification. First, we have simple, that is contain only amino acid, and we have conjugated. We have simple proteins, that is a protein that only have that only have amino acid in them, and we have some proteins that are conjugated. That is, they are simple protein but with prosthetic group attached to them. Then we also have level of structure of proteins. So the level of structure of proteins started from primary structure, secondary structure, and then tertiary structure, as well as the uh, quaternary structure. So the order of amino acid in a polypeptide chain reads from the N-terminal end to the C-terminal end. So the L N-terminal end to C-terminal end, we have the primary structure. Like for example, in the case of primary structure, we just have amino acid starting. Of course, remember we have amino acid that usually linked together using a peptide bond from our previous explanation. So this is how the linked 1, 2, 3. Look at it. We have, this is how the linked. So one amino acid linked with another amino acid through a peptide bond. So actually, the order of the amino acid sequence in a polypeptide chain, it's usually read from N-terminal. So this is started from N-terminal. So the left is the N-terminal, while the right is the C-terminal. So it's just the primary structure. In the primary structure of amino acid, it's just you have the what you call the sequence of the amino acid. The sequence of the amino acid is what actually determines the primary structure of the proteins. And generally, the primary structure of the proteins is what determines the protein folding as well as the protein function. And then the next thing is the secondary structure. So in the case of the secondary structure is the rearrangement, or is the arrangement in space of a backbone atoms. Secondary structure examples, we have alpha helix and beta belated sheet. So in the case of secondary structure, it's just that from the primary structure, they are, remember in the peptide bond, we have some pre-amines and some free carboxylic hydrogen. So what they use to, what actually happens is within that same polypeptide, that free amines and carboxylic group will now link together with some additional hydrogen bond. That is how we have the secondary structure. But in the case of tertiary structure, is a 3D arrangement of atoms, including those in a side chain and prosthetic groups. So in this case of the tertiary structure, remember we have non-covalent forces. So whenever we said non-covalent forces, we have so many examples of non-covalent forces. What are the examples of non-covalent forces? We have hydrogen bond. Hydrogen bond is an example of non-covalent force, non-covalent bonds. And then apart from this, we also have van der Waals. Van der Waals, van der Waals force of attraction. And apart from the van der Waals hydrogen, van der Waals force of attraction, then the third one, we have also electrostatic force of attraction. So all these are non-covalent bonds. So the hydrogen bonding and as well as van der Waals force and electrostatic force of attraction of the secondary structure of proteins is what actually brings the tertiary structure of the proteins. So the tertiary structure of proteins is due to further bonding, which hydrogen bonding, bondal force of attraction, as well as electrostatic force of attraction. But in the case of tertiary structure of the proteins is when you have actually a several polypeptide proteins change with multiple subunits and then they will now linked up together to produce what we call a protein complex. And that is what we call the quaternary structure of proteins. Okay. So now I think we are also, okay, I think I'm going to stop here. I'm going to also upload more other videos. Thank you.